Hi, everyone. We are Callen Bragdon, Mengzi Lee, and Rebecca Miller from Boston University. The project we are presenting is called SigRepo, a structured platform for the storage and streamlined analysis of multi-omics signatures. This project was funded by the NIA, the National Institute on Aging. In recent years, generating high-throughput omics data has become standard in biomedical studies. These omics datasets encapsulate different features, such as genes, proteins, or metabolites. Many of these datasets are stored as raw data in publicly available repositories, such as the NIH's Gene Expression Omnibus and ArrayExpress. Our recurrent goal is to extract from these datasets differential signatures, that is, lists of up and down regulated features associated with study-relevant phenotypes. However, extraction of these signatures requires considerable processing and domain knowledge. What's more, if your lab is constantly generating many signatures, it can be difficult to keep your data organized efficiently for easy access. Here, we present SigRepo to address these problems. SigRepo consists of three layers: the data layer, a middle layer, and a front-end layer. The data layer consists of a relational database built using MySQL and a file system storing instances of an R6 object called omics signature. This omics signature object was designed to efficiently process and represent signatures in R. Our middle layer contains a Python Flask API designed to accommodate access-controlled, programmatic retrieval of signatures. The front end, or application layer, is an R-Shiny web interface that connects to our database and performs basic analysis. Our application will be deployed as a bioconductor package, installable on most computer architectures. Users will be able to have their installation point to their own database to allow for creation of internal management systems. Alternatively, they can point to our publicly hosted database and have access to a much wider repository of signatures collected from many sources. While we created the R-Shiny interface for ease of access for users with any level of familiarity with the tool, all functions used on the web interface are also command line accessible from the R console. The main features include the following: signature search, signature comparisons, and gene set enrichment analysis using the HiTSeq package, all of which will be elaborated on later by Mengzi.
Now I'm going to introduce you the omics signature object and our R-Shiny interface. To store signatures efficiently, we created the R6 object omics signature. Omics signature contains metadata and a multi-resolution representation of the signature information. This includes the differential analysis table, if available, as well as a feature symbol list, generally a list of significantly differentially expressed genes or proteins, optionally weighted by a test statistic. The omics signature object has several built-in methods, such as extracting a new signature according to new criteria, and creating an omics signature collection object to store multiple related omics signature objects. You can see our R-Shiny interface design at the bottom of the poster. For signature search, you can select search terms from each of the drop-down menus. In order to make the search more user-friendly and efficient, each drop-down menu is automatically updated according to the other terms you selected. After clicking the search button, the app will display a downloadable table of signatures matching your query. On the compare page, you can select two signatures from the database and see the overlap of the two signatures, as well as perform a KS test to compare to ranked signature lists. We also incorporated the HiTSeq package to perform gene set enrichment analysis. After selecting a signature from the database, you can select gene sets available from SigDB, or upload your own gene set list for performing the enrichment analysis. We also have a user control feature as shown at the bottom left. There is a login tab on the interface, and logging in allows the user to upload new signatures to the database. If the logged in user is an administrator, they will have access to additional pages for updating the database, such as adding new SC platforms or features. To wrap up, we created a management and analysis platform capable of efficiently and conveniently storing signatures generated by high-throughput omics studies. In addition to storing these signatures, our application can also conduct some basic analysis on both individual signatures and pairs of signatures. In the near future, we will apply this platform to analyze signatures of centenarians, which means people who live at least 100 years. Furthermore, we intend to integrate more analysis features to this platform to make it a powerful tool in omics analysis.